Thanks for the introduction. Hello, everyone. I'm Qiuying Liao, currently a final year PhD student in the University of Hong Kong under the supervision of Dr. Jenny Lan. Today, I'm happy to share about my research project, Inhalable Powder Formulation of a Broad-Spectrum Antiviral Agent for Respiratory Viral Infections. So as we all know that respiratory viral infections are posing significant threat on global health, especially most recent COVID-19. Effective antiviral agents are essential for infections treatment. However, the development of specific antiviral or prophylactic vaccine for new emerging virus can take years, and it may lag behind rapid expansion of the pandemic. So the early empirical administration of a broad-spectrum antiviral will improve the patient outcomes and facilitate the control of the epidemic before a specific antiviral is developed. So a recent study reported that a retinoid derivative AM580 shows broad-spectrum antiviral efficacy by targeting the cellular lipid metabolism. And it was demonstrated that AM580 has the activity against MERS coronavirus and influenza virus. And that study also reported that an analog of AM580, another retinoid derivative, as shown on the right-hand side, it's called tamibarotene. And it also demonstrates it has in vitro antiviral activity. So tamibarotene is available in the market and as oral tablet for the treatment for leukemia. And in order to allow maximal drug concentration in the lung, reformulating tamibarotene for pulmonary delivery, and so that it can be directly delivered to the primary site of infection and with easy administration. But due to the very low water solubility of tamibarotene, here we use secodation to improve the solubility. And compared with other pulmonary dosage form, dry powder formulation are more stable as it is in solid form and more suitable for high dose loading. So in this study, spray freeze drying technology was used as it can produce porous particle which has lower density and therefore smaller aerodynamic diameter, so that it will be suitable for inhalation. And also the porous structure can also increase the surface area and therefore to facilitate the dissolution. So basically, spray freeze drying is a two-step process. The liquid was atomized into droplet and get frozen immediately in a cryogen. Usually we use liquid nitrogen. And then the frozen droplet was freeze-dried under low temperature and pressure. And in this formulation, we use butyl alcohol as a co-solvent for tamibarotene. Also because it has a relatively high freezing point to allow sublimation without extremely low temperature. And this slide shows the morphology and aerosol performance of the spray freeze-dried tamibarotene powder. So as shown in the left panel, the unformulated tamibarotene display roll-shaped crystalline structure with long and heterogeneous lengths, whereas the spray freeze-dried powder are spherical and porous. And the geometric size is less than 10 micron as seen from the SN image. The in vitro aerosol performance was evaluated by NGI coupled with a breathaler. And we can see that the emitted fraction was more than 90%, and the fine particle fraction was higher than 60%. And the MAD was around 1.8 micron within the inhalable range. So this all indicate that the spray freeze-dried tamibarotene powder has a satisfactory aerodynamic property. And next, we evaluate the dissolution profile of the powder. As the fine particle dose, which has an aerodynamic diameter below 5 micron, are expected to deposit in deep lung region. So only the fine particle dose was separated by an FSI and for the subsequent dissolution study. So as shown in the right-hand side, due to the very low solubility of tamibarotene, the unformulated drug dissolved in a very slow pace. And whereas the formulation show rapid dissolution profile, and this could be an this could be a combined result of the solubilization of secretion and also the enlarged surface area brought by the porous structure. And further, we move to in vivo pharmacokinetic study in the healthy mice. So we use healthy biopsy mice for the PK study. And this two figure shows the curve of drug concentration in plasma as well as the lung versus time after intratracheal administration of the powder and after intraperitoneal injection of the unformulated drug. So for IT group, for both plasma and lung tissue, the drug concentration reached a peak at 5 minutes after administration, and which is consistent with the rapid in vitro dissolution rate. So the table shows PK parameter after IT and IP administration. So we can see for IT group, it results in higher Cmax, the maximum concentration, and higher IUC, so which suggests the pulmonary delivery of the tamibarotene powder can achieve higher bioavailability in both pulmonary region as well as the systemic circulation. And with the enhanced bioavailability, now we are currently working on the pharmacodynamic aspect. So the antiviral study is still ongoing. So far, we have demonstrated that in vivo prophylactic protection effect of the tamibarotene powder against influenza A virus H1N1 and the MERS coronavirus. And the study of the in vivo antiviral effect against SARS-CoV-2, which is the cause of COVID-19, is still ongoing. And that would be all I want to share today. I would like to acknowledge the funding from the University of Hong Kong. And I would also want to thank my supervisor, Dr. Jenny Lam. We also like to thank our collaborator, Professor Qiuying from the Department of Microbiology in the University of Hong Kong, and all my lab mates for their assistance in this project. And thank you all, and all questions are welcome.
Thank you very much. If I can just start with a question, may I ask, do you know what the optimum lung pharmacokinetic profile would be based on the pharmacology? Do you need a high peak concentration, or do you need a long duration of drug in the lungs?
And thanks for the question. So usually the efficacy of the antiviral activity is concentration dependent. So of course we expect a high drug concentration in the lung region for its effective antiviral activity. Yeah. So.
Yeah. And a number of questions around the use of cyclodextrin and the dissolution. So could you alter the cyclodextrin to change the dissolution profile? And then some questions around the safety of cyclodextrin and how you perform the dissolution assays.
Sorry, I cannot.
Yeah, sorry,
hear
it's three
the
questions in one. I'll break them down. So first, you use cyclodextrin.
Yes.
Could you adjust the amount of cyclodextrin to change the dissolution of your formulation?
So so far the ratio between drug and cyclodextrin is the mass ratio is around 1:9. So in the formulation it contains 90% mass ratio of cyclodextrin. And so far we haven't. We will investigate how the amount of cyclodextrin will affect the dissolution rate, and we will also look into how the different type of cyclodextrin will affect the aerosol performance as well as the dissolution profile of the formulation.
Okay, and the second question is, what did you use as a dissolution medium? Was it a biorelevant dissolution method?
Thanks for the question. And what we use is water, but the volume is 100 milliliter. So we didn't use a biological related medium at this stage. Yes.
Yeah, and do you know how safe cyclodextrins are in the lungs?
So we will perform the toxicity study, such as the histological study, to investigate the side effect of the excipient on the animal in the upcoming study. And so far, based on the literature search, and there was the safety of secretion on the pulmonary delivery is the toxicity is there's only a few papers that reported the toxic effect of secretion for pulmonary delivery.
Okay, thank you very much. We will forward any
Thank you.
to you.
Thank you.